everyone! Today we'll be talking about negative single-stranded RNA viruses. These refer to bad prof, Bunya, Arena, Delta, Paramyxo, Rhabdo, Orthomyxo, and Philo. They are Baltimore class 5 viruses because they are negatively single-stranded and they need a viral RDRP, RNA-dependent RNA polymerase, to make a positive single-stranded RNA that is complementary to the negative single-stranded RNA and is exactly the same as mRNA, so that it can be used for the translation of proteins. And it carries this RNA-dependent RNA polymerase in its virion because you can't make proteins without this. Additionally, it has an envelope structure, which basically means that you will enter via receptor-mediated endocytosis or direct fusion and it is also a helical structure, so unlike positive single-stranded RNA, it is unlike positively single-stranded RNA, which is icosahedral. And you can analyze them in two ways. They can have a segmented genome or a continuous genome. Segmented genome means there's several RNAs in the genome, and these refer to BAL, or Bunya, Areno, and Orthomyxo within this family. You can remember this as a segmented bun. One specific example we're going to look at is the influenza virus, which belongs to Orthomyxo. Influenza has about 8 segments, and these 8 segments encode for 8+ different proteins, each mRNA encoding for a different each mRNA coded by a different segment, which then codes for a protein. However, for the M1 and NS1 protein, which are encoded by 2 segments separately, they can be further spliced using host machinery to make M2 and the NEP protein, which is important in the matrix and capsid respectively. And this splicing machinery is only found in the nucleus, which is why all of this replication takes place in the nucleus, unlike the other RNA viruses, which all take place, its replication, in the cytoplasm. Now, of these 8 different proteins, 8+ different proteins synthesized, we need to remember PB1, PB2, and PA, because these proteins come together to form the influenza RNA-dependent RNA polymerase, or the ribonucleoprotein complex. And this is assembled again in the nucleus and organized in the nucleus. So their functions include, PB1 binds this 5' terminal sequence of negative single-stranded RNA, and then by doing this, the PB2 can be activated to bind the 3' end of the negative single-stranded RNA, and then this obviously begins the transcription of positive single-stranded RNA from negative RNA, and then the PA part comes along, which is an endonuclease, and it cleaves the host mRNA from its 5' cap, and then grabs this cap and puts it onto the newly synthesized viral mRNA. And this cap, or the M7 guanosine cap, it acts as a primer for host translation factors to bind and initiate the translation of viral genes rather than host or cellular genes. None of this can occur though if the host doesn't transcribe mRNA and add the cap onto it using methyltransferase. And the only reason why influenza does this is because it doesn't have that methyltransferase to add that cap on. So it just decides to steal. Now moving on to the unimolecular genomes. So these usually have many mRNAs that are encoded by the one genome. And these mRNAs arise because there's intergenic termination sequences and polyase signals between the different types of protein genes. And this basically tells the viral RDRP to stop for a minute, and sometimes it causes the viral RDRP to fall off, which means that, this occurs about 20% of the time, which means that proteins, genes which are located near the 3' end of negative single-stranded RNA are synthesized first, because transcription occurs in a 3' to 5' direction along negative single-stranded RNA. And this also means that because the nucleoprotein is located there, it is in the greatest quantity for synthesis. And it is in the greatest quantity for synthesis because it has important structural roles. First in the nucleocapsid formation, then again in the ribonucleoprotein complex, or the RDRP formation. Additionally, it can bind to the intergenic sequences we previously discussed, and this just tells the RDRP to be more stable and continue replicating along the negative single-stranded RNA to copy the genome, rather than just prepare a bunch of mRNAs from that. These are used by Delta, Paramyxo, Rhabdo, and Philo families. And now we're going to look at how these negative single-stranded RNA viruses, which have a continuous genome, make so many different proteins despite its simplicity, using the Henipa or Hendra virus as an example. So, like the previous mechanisms we discussed, it uses the imperfect scanning of negative single-stranded RNA to produce, different types of mRNAs, which then code for different types of proteins. And this mRNA, where it skips and is kind of imperfect in its copying, is called the P protein. And this P protein, is important for the RDRP, but when it doesn't scan properly, it can make the V protein or the C protein, and both of them play a role in inhibiting interferon alpha and gamma beta responses, which again enhance viral survival. The last category is a double-stranded RNA virus family called the Reoviridae family. These include the rota and orthoreoviruses. Because it is double-stranded RNA, it is highly immunogenic, and there's about 10 to 18 segments of double-stranded RNA within the Reoviridae family, but it's able to hide from host immune defense very well because it has about 3 layers, and deep in its core is the double-stranded RNA. So it is hidden away, and this core is wrapped away in ISVP and then wrapped in the virion. Upon entry, the virion undergoes proteolysis and then releasing the ISVP core, but then the ISVP core then releases the core. But even in the core, you can't detect that double-stranded RNA, and then that's where its mRNA replication takes place in vitro. Okay, thank you so much for listening about the negative single-stranded RNA. I think they're very interesting, and I hope you find them interesting too. Bad prof eats segmented bun.